Dear students, in this video lecture we are going to study structure of male reproductive structures of plant called anther. So let's begin. As we know, in flowering plants we have two different types of whorls. One is reproductive whorl and other is accessory whorl. Accessory whorls include calyx and corolla, whereas reproductive whorls include androecium and gynoecium. Androecium is the male reproductive part, which consists of individual units called stamen. And the stamen is having two different parts in it. One is filament and the other is the anther, which is present at the tip of the filament. This filament is connected to anther by a tissue called connective. Typical anther contains two anther lobes. As you can see in the previous slide, it was having two anther lobes which are connected with connective, and in each lobe we are going to find two pollen chambers which are considered as microsporangia or pollen sacs. There are two kinds of anther based on the lobes, number of lobes they contain. If the anther contains a single lobe, such anther is called as monothecous anther. The example of monothecous anthers are Hibiscus rosa sinensis, and normally the anthers contain two lobes, and such anthers are called dithecous anthers. Example is citrus. Pollen grains which contribute male gametes are present in each microsporangium, or we can say are formed within the anther. If we take a cross section of mature anther, you can clearly see different regions, like the outermost region of the anther is made up of single layer of cells called as epidermis, which is followed by a layer called endothecium, and endothecium is followed by 2 to 3 layers of cells called middle layers, which is followed by the innermost layer called as tapetum. Tapetum is considered as the protective layer for every microsporangia. So inside the tapetal layer, we see the development of microspores from the microspore mother cell. So during the development of microsporangia, when we take a section of transverse section of young anther, we can find the undifferentiated mass of cells. Later on, some cells will attain the greater size and they differentiate to form archisporial initials. These archisporial initials divide and give rise to two different kind of cells: primary parietal cells towards the outer side and primary sporogenous cells towards the inner side. Primary parietal cells are further responsible for the development of parietal cells and an endothelium, whereas primary sporogenous cells, they are going to give you microspore mother cells. They differentiate and give microspore mother cells or pollen mother cells. Microspore mother cell is diploid in nature, which undergoes meiotic division to produce four pollen grains. And these four pollen grains remain attached with each other, hence they are named as pollen tetrad. These microspores are released outside by the breakage of endothelium layer. Endothelium layer plays a very important role during the dispersal of pollen grains through the area called as stomium. As they mature, the endothelium cells are going to dry up and it creates a pressure on the stomium to break and release the pollen grains outside. Thank you.